Good evening, everyone, and welcome to Maria Mitchell Association's January Science Speaker Series. And tonight I'm privileged to host Dr. Elizabeth Sherman. She will be providing an update on the Maria Mitchell Buoy, emerging stress indicators and cross-platform integration. And I did not introduce myself. I'm Janelle Gurley. I'm the Director of Science and Programs at the Maria Mitchell Association. And before we hand it over to Betsy, I'll just give us a quick overview of what Betsy will be sharing about tonight, as well as her extensive and very accomplished bio. So during our presentation this evening, Dr. Sherman will share an update on highlights on what high-frequency buoy data reveals, short-lived events, repeated excursions beyond biological thresholds, and the sequencing of changes across oxygen, temperature, pH, chlorophyll, salinity, and nitrate, patterns that are often invisible in grab samples collected days or weeks apart. She will also summarize progress on cross-platform integration, enabling buoy data to be compared with other local monitoring efforts here on Nantucket that are used to flag conditions that warrant closer attention and guide future questions. Dr. Sherman is the Maria Mitchell Association Senior Marine Science Advisor, a role that she assumed last summer. In this position, she has shaped the MMA's Nantucket Harbor Buoy program into a rigorous, decision-relevant monitoring effort focused on harbor biodiversity. She established verification procedures for continuous sensor measurements, cross-checked buoy data with other regional monitoring records, and strengthened data collaboration with partner organizations. She also advises on program design and provides public outreach talks that translate sensor records into ecological meaning. Dr. Sherman's work leverages continuous buoy data to address ecological questions that grab sampling rarely resolves, including how often conditions cross biologically meaningful thresholds, how short-lived events accumulate, and how multiple sensors interact across temperature, oxygen, pH, and other nutrients. Her analyses have identified recurring temperature patterns consistent with sublethal stress, clarified nitrate dynamics in collaboration with Nantucket's Natural Resources Department, and kept pH research focused on impacts to eelgrass, bay scallops, and the broader harbor community. Dr. Sherman is Professor Emerita of Biology at Bennington College, where she spent more than 4 decades teaching and conducting research. Her scholarship focused on environmental physiology, evolution, and behavior in freshwater and marine systems alike, with a long-standing emphasis on coral reef biology. Her peer-reviewed publications span thermal tolerance and climate stress, amphibian physiology and disease, and coral reef dynamics. She also held a long-term appointment as visiting scientist with the Cayman Islands Department of Environment, and founded several field-based learning programs, including the Bennington College Coral Reef Project. And Dr. Sherman earned her BA in biology, high distinction from the University of Rochester, a PhD in zoology, high distinction from the University of Vermont, and completed postdoctoral research in neurobiology and behavior at Cornell University. A central theme throughout her career has been strengthening public understanding of science and its role in civic life through education consulting, and extensive public speaking across the New England area. Her work has been supported by numerous awards and grants, including multiple grants from the NSF or National Science Foundation. Join me, everyone, in welcoming Dr. Sherman. Betsy, it's such a pleasure to have you back. Welcome, welcome.
Thanks, Janelle. I can't believe you failed to mention my Academy Award nomination, but
I'm write that one down for next
time. I'm really thrilled to be here to talk to you all about what we've been finding using the data from the Maria Mitchell Buoy. Typically, when I give a talk, I like to see my audience. I like to do it in person so that if I'm laying an egg, I could maybe improvise and pull up the energy a little bit, but that's not possible tonight. So we'll see how I do in this venue.
You're going to do great. I turn it over to you, Betsy.
Oh.
Nope, you're all good. You are good.
Did people hear anything I just said, or?
Yes, they did. They heard everything you just shared.
All right. So when I was rereading the blurb I sent to Maria Mitchell to note what I was going to be talking about tonight, I realized I omitted something really important, and that was the big idea. As I'm sure you all know, we humans are doing quite a bit of damage to our ocean systems. The work of the buoy and those of other monitoring programs in Nantucket uh, is really important for the health of Nantucket Harbor. But more than that, it is a part of our understanding of global oceans. The Nantucket Harbor is an important component of that huge system. And to that extent, I'm going to dive into the weeds with us for a bit from time to time. But then I'm going to pull us back to remember that, hey, this is part of a much larger system. So here's the buoy, and it's gorgeous and it's solar powered. And this is just a map to orient us as to the position of the buoy in its deployment. This is the southern part of the Cape. Here's the vineyard. Here's Nantucket. And the buoy is right there. It's positioned right at the mouth of the harbor. And as you might imagine, with this buoy, it is anchored in this position in the harbor, so it doesn't move. Uh, but I think of its location as a kind of uh, best reference point because, for example, if the oxygen content of the water around the buoy is low, what might that mean for other more sequestered embayments and coves? So, if the buoy is sitting still and only giving us data from one position, we have also the monitoring that the Nantucket Natural Resources Department is doing, and they report their data in a site called WQAV. You can find it on their website. And while the buoy is anchored here, the monitoring program of the NRD has a lot of spatial information. So they take readings of various chemical and physical properties of Nantucket Harbor in different places within the harbor. So the buoy gives us high frequency data every 15 minutes. These samples by the NRD are episodic, they're discrete samples, and we can use both of these monitoring programs to complement one another. So keep in mind the buoy is recording and transmitting high frequency data. Every 15 minutes, it is logging data on these kinds of sensors. The buoy sensors include one for temperature, pH, dissolved oxygen, chlorophyll, salinity, and there's another sensor for nitrate. Now, these sensors are extremely sensitive and very responsive, but they are also quite vulnerable to various perturbations. And one of the things that's been important for MMA to do is to learn how sensitive they are and to look at our data and see which data are reasonable and not. And I'll talk a little bit more about that. But so we're going to get all of these data every 15 minutes during the deployment of the buoy, to which I ask, so what? The numbers in and of themselves are not particularly interesting. What is it that we really want to know? We want to have some measure of the health of the harbor. How is the harbor doing? So if I say, well, the pH is 8.1, so what? It matters. The 8.1 only matters insofar as it affects the organisms in the harbor. And that is our ultimate measure of harbor health. How well are the species doing? What is the composition of the various communities? Uh, uh, are populations persisting? Are they growing? So our measure of harbor health is how the key species are doing. Now, remember, I said we want to correlate how the species are doing with data from the buoy. So in order to do that, we have to be really certain of our buoy data. So I want to just give you a quick and dirty of how we look at these data and decide which are real and which aren't. So this example that I'm giving you is from the chlorophyll sensor. And I'm going to take a little drink here. These were the first data from the buoy that I downloaded uh, and had them plotted. And I just was kind of stunned when I first look at these data. So, just to orient you, this is a time series and shows that we collected data from August 2024 into November of 2024, and then again in May of 2025 into the fall of 2025, which we don't see here. But when I first looked at these data, I said, well, my goodness, there are a bunch of days where the chlorophyll reading is minus 1,000, as though somehow the harbor were owed chlorophyll. And these levels, way up high here, of 600 micrograms per liter, are absurd. I don't think a spinach smoothie would have that kind of data, that kind of level. So my first uh, learning experience was to get rid of these placeholders and absolutely absurd, impossible anomalies. So my second pass through the data, and note now that my axis makes more sense going from 0 to 50. But I wondered about these data above 30 micrograms, and here's where I have to consult the literature to see what is reasonable for Nantucket waters. And with conversations with other scientists and my own research, levels of chlorophyll above about 30 would suggest really impossible bloom levels for Nantucket. So I removed those data too. My third pass through these data is to compare the chlorophyll data, which are indicated in this top trace here, with data from the other sensors that were deployed simultaneously to see if data up to about 30 micrograms are plausible. So when I put these data into ecological context, I would ask myself, well, look at these high chlorophyll data. That might indicate there's a lot of photosynthesis going on. But I don't see anything comparable, for example, for the dissolved oxygen buoy. I don't see anything comparable in the pH sensor, sorry. So I can refine what I include as legitimate data using the understanding of ecology of the harbor. And the last QA, QC, did I mention that's what we call it? Quality assurance, quality control. So the last part of the quality control is to compare the buoy data. Here is September, October, and November of 2024 with other monitoring systems. And in this case, it's the WQAV data from the NRD. And here you can see there are these discrete samples in March of 2024. And when you look at the data where both monitoring programs overlap, you can see that there's very good agreement. So I am confident that it is legitimate for me to remove data above about, uh, excuse me, looking at my notes. Don't do that. I'm confident that these data are legitimate data. So what we want to do is think about what the numbers tell us and why they're important. So what we're going to do is go through a few case studies. I'm going to describe the problem and what data we're going to use to interrogate the problem. I'm going to give a short QC quality control of the data so you're persuaded these are legitimate data. And then, of course, what do these data mean? And there will be a short quiz at the end of this talk. So let's take this buoy out for a spin and see what we can learn. The first problem I want to discuss is eutrophication. And eutrophication occurs when waste and fertilizer runoff and leaching and storm water dumps materials into the harbor. And among those materials, some of them can actually serve as nutrients for algae. And one of the consequences of all of this dumping of material into the harbor is that there may very well be what are called algal blooms, in which a ton of algae grows very, very quickly. There are consequences to that. When uh, the algae become so numerous up here, they can form mats and they can actually block sunlight from other plants that are essential for the harbor, including eelgrass. They can block the sunlight from the eelgrass, impairing the eelgrass capacity to photosynthesize. And the other thing that happens is these algae put out a ton of waste. They die and sink to the bottom. And there are decomposers, bacteria mainly, in the harbor water that break down this decaying matter. And these decomposers use oxygen in order to break down their food, the waste of the algae, in the same way that most organisms on this planet use oxygen to metabolize their food. And as the bacteria use up the oxygen content of the water can actually decrease, and a lot of animals can suffer as a consequence of that. This is an aside, but when I learned that oxygen was simply a waste product of photosynthesis, it blew my mind. And before there were photosynthesizing organisms on the planet, the cyanobacteria and algae and plants, there was no molecular oxygen on planet Earth. That came about 2.5 billion years ago. So, roughly half the lifetime of our planet was without oxygen. And of course, the advent of an oxygen atmosphere on our planet led to the evolution of the extraordinary diversity that we see today. Okay, so what can we measure to try to interrogate eutrophication? Well, obviously, nitrogen is a reasonable a substance. The concentration of that is reasonable to look at for interrogating uh, eutrophication. And the measure of nitrogen that we use with the buoy is nitrate. This is a lousy slide, but it's mostly to remind me to tell you that the scientific literature has established various levels of these different variables we're going to talk about and how they might affect the organisms living in the harbor. So they can indicate when eelgrass and bay scallops and fish might be in worrisome conditions. But of course, a picture's worth 1,000 words, and this is beautiful, healthy eelgrass habitat. This is not. And try imagining the eelgrass underneath getting sufficient sunlight to be able to photosynthesize. By the way, I took this from the town of Nantucket water quality page. So here is a time series of nitrate in that we derive from the Buoy data. Here is the deployment in 2024 and the deployment in 2025. Now, uh, nitrate is not the only component of nitrogen in the harbor. There are other components of what's called total nitrogen in the harbor. So initially, here's part of the QA/QC. I compared the nitrate levels from the buoy to the WQAV data, these discrete samples taken by those occasional monitoring trips taken by the NRD. And what you can see is that the nitrate levels established by the buoy are roughly 20% of the total nitrogen in the harbor. And I kind of went, eh. And I looked in the literature, and the suggestion was that 20% is awfully high as a component of total nitrogen. So the next thing I did was I looked at comparing actual nitrate data with the buoy data, and later in the Q&A, if you want, I can talk about what that designation means. But as you can see, yeah, this nitrate data seems lots higher than the nitrate data from the buoy. I also went into the NRD data and looked at WQAV data. And what this graph shows is the various components of total nitrogen. And obviously, the highest dot is going to be the total nitrogen. But these are these are most of the components of nitrogen. And you can see this yellow ball, DIN, dissolved inorganic nitrogen. Its component is way down here. So this is where the nitrate would be. Um, and this suggests that the buoy data are in fact elevated for whatever reason uh, compared to the NRD data. By the way, there is a threshold established for what becomes problematic for the eelgrass, and it's at about 25 micromolars. So while we're not there in these discrete samples, it's certainly something to continue to watch for. So let's go back to the buoy data. It's likely, as I said, that these specific values are elevated because of calibration, drift, fouling problems. And in conversation with Janelle Gurley of MMA and also Tara Riley at the NRD, we figured out a way to be able to calibrate the Suna nitrate levels, calibrate the sensor more effectively next summer, so that uh, we can rely on those data more robustly. But what I wanted to know in looking at this past year's data is, can we use short-term changes, not the absolute values, but short-term changes in nitrate uh, to see if there can be signals we might pick up to serve as an early warning predictor of eutrophication. So keep in mind, nitrate is not a large component of total nitrogen, but the event signal can be very, very quick in the way of hours or days, and the buoy is certainly able to accommodate that. So, in thinking about how I might determine whether the nitrate levels in the uh, in the harbor, or actually the changes in nitrate levels in the harbor, are sensitive enough to warn about eutrophication. I had a conversation with Charles Johnson, the stormwater guru of Nantucket, and he suggested that I look at rainfall data and see if there were any spikes in nitrate levels uh, as a consequence of rainfall in the harbor. And so I downloaded data from NOAA that covered the same deployments of the nitrate data. And I had two hypotheses as to what to look for relative to rainfall. The first is that rainfall might cause a decrease in nitrate due to dilution, that is, fresh water coming from the rain. The second hypothesis, more along the lines of what Charles was thinking about, is that rainfall might cause an increase in nitrate due to runoff from the land. So, uh, I wanted to really uh, make a high bar for rainfall so that I could see a significant effect. So I decided to look at uh, 6 rainstorms above 1.5 inches in rain and compare that with the nitrate buoy data. So what I've plotted here is on the y-axis is the net change in nitrate relative to the day of the storm. So this shows nitrate levels relative to the day of the storm, whether they are increasing or decreasing. This is after day one. This is 2 days after the storm, and this is 3 days after the storm. So for most of the storms of 1.5 inches or greater, there is a measurable increase in nitrate. So I thought, ooh, that's interesting and not unlike what Charles was predicting. But then I went into the data deeply and I looked at within storm day-to-day trajectories and found that, for example, a rainstorm of 1.76 inches on the first day, the nitrate level went up. On the second day, relative to the first day, it went down. On the third day, relative to the second day, it went down. So using that day three average, I thought might be a blunt instrument. Um, so again, I had a conversation with Charles, and he suggested that perhaps I look at tidal data. So again, I downloaded tidal data from NOAA, and here are the tides, and I've matched it with the nitrate data, and this is a week in September of 2024. And Charles' question was, well, maybe the tides are simply bringing the same mass of water uh, into the buoy and out of the buoy, back and forth across the buoy. So, as my proof of concept, I also looked at the salinity data to see if the sensors were sensitive enough, in fact, to change values as a consequence of the tide. And the salinity data certainly show that. So when there's a flood tide and ocean water is zooming in, you see the salinity go up. When the tide goes out, you see the salinity going down as fresh water from the land fills in. And these match quite neatly. The nitrate levels don't follow those changes as precisely as the salinity data. So uh, we're still looking. What I'm searching for is I'm trying to separate signal from noise. That is, are these little peaks, are they significant, or is it just this sort of noisy variation that we see from the nitrate? And secondly, if these peaks are significant, does the magnitude of these changes actually affect the critters in the harbor, notably the eelgrass? So to be continued. That's what I'm going to continue to look at with partners from other monitoring programs in Nantucket. Uh, Back to eutrophication, there are two other variables that we can measure. We can measure the chlorophyll content of the water because as the algae increase, they will be revealing greater concentrations of chlorophyll. And the other thing is the oxygen depletion that is caused by the decomposers chewing up the dying algae. So those are two other variables we might check. And uh, here again, that boring slide, just to know that there are levels of chlorophyll in the literature that warn us of, say, mild enrichment or early shading of eelgrass at levels above about 0.5 micrograms per liter. A little more QA/QC. Remember that the chlorophyll levels from the buoy match very nicely the chlorophyll levels taken from those discrete samples provided by the the NRD. Same with dissolved oxygen. We can see that there are various levels of oxygen concentration that have more or less consequences for the organisms in the harbor, notably the bay scallops and the fish. And here I do my QA/QC thing where we look and see that the uh, WQAV, the discrete sampling data, match the oxygen data from the buoy, so we're good to go. And I'm going to use chlorophyll as a proxy for algae and the dissolved oxygen as a measure of hypoxia. Can these two variables provide an early warning prediction of eutrophication? So what I've graphed here, and this is from a week in May of last year, the blue trace is the dissolved oxygen. I've also included these shaded bars representing nighttime from about 11 p.m. to 4 a.m. And what you can see from the oxygen trace is very predictable. During the day, oxygen content goes up because of all the photosynthetic activity of the plants and algae in the harbor. And then at night, whoops, when everybody is respiring, using up oxygen to metabolize, you see the oxygen content go down. So this is a perfectly understandable daily cycle or diel cycle of oxygen content. The chlorophyll a little bit trickier, and but in general, during the daytime, we see chlorophyll A increase, which is what we would predict. The lack of absolute following with oxygen is something we can talk about in the Q&A if anyone is interested. But there is no obvious creeping up of the baseline chlorophyll data, nor a following oxygen depletion. But I would argue that these two sensors on the buoy are sensitive enough uh, to be able to detect conditions that might warn us of impending eutrophication. Another problem, warming seas. Global warming is uh, absolutely confirmed by scientific studies from all over the world. The sea temperature is changing as a consequence of human behavior, and why our present government is uninterested in it is problematic to me, but I digress. These graphs are representations of what could happen to individual organisms as the temperature increases, and as a consequence of what happens to the individual organisms, what happens ecologically to the abundance of these organisms. So what is very common is that increases in temperature, really high increases in temperature, cause decreases and worrisome changes in physiological function, whether it's how quickly we're uh, respiring, how quickly our proteins are able to do their functions in our cells. And the problems that causes for individual organisms is, of course, reflected in the abundance of those organisms. So as the temperature of seas increases, we see the abundance of various species decreasing. And these phenomena are well documented in the scientific literature. So, obviously, the sensor that I'm going to use to interrogate this is the temperature sensor. Here's that uh, kind of unpleasant slide, but I do want you to pay attention to this value of uh, when the literature says early stress might have an impact on the organisms in the harbor at temperatures above 23 degrees Celsius or 73 degrees Fahrenheit. At those temperatures, organisms might begin to see some sublethal stress. Here's the QA, QC, where we see that the NRD data match the Buoy data, so we're all set there. And I also plotted the Buoy data are in blue dots and NOAA data are in red. So you can see that we have quite a bit of legitimacy in the Buoy data. So my question is, does temperature uh, act as a sublethal stressor on the organisms in the harbor? Now, at least where the buoy is moored, the temperatures aren't going to get up so high that organisms just conk right out. But there are, remember that 23 degrees, there are temperatures that are not lethal, but that still can have deleterious effects on the organisms in the harbor. So here is the time series for temperature, and here's the November, or I'm sorry, the 2024 data. Starting in the summer, obviously the temperatures go down, and the 2025 data, obviously from May into the summer, the temperatures go up. And I've also included that 23-degree level threshold. Uh, and what is clear is that the organisms in the harbor are already experiencing sublethal temperatures. So, uh, you know, got my attention. Now, we can quantify not just when the temperatures are above that 23 degrees, but also how many hours additional exposure are the organisms getting. And this brings up an index that we call the daily degree hours. The degree hours capture both the intensity and duration above a threshold. And here I've set the threshold at 23 degrees. And each bar uh, is the sum of all extra heat that day. And by extra heat, we mean how many hours above 23 degrees. So you can see, just as there were little bits last in 2024 and a larger episode in 2025, you can see that here are the sublethal stressors in the last year, in 2024, and here in the summer of 2025, the organisms were really exposed to some significant sublethal stressors. And keep in mind that the position of the buoy is in a well-flushed part of the harbor. So if these are the daily degree hours above 23 degrees in a well-flushed portion of the harbor, what might be going on in those more sequestered embayments and coves? The last problem I want to discuss is ocean acidification. Many of the organisms in the harbor require calcium carbonate to be precipitated out, whether it's a part of their exoskeleton, whether it's a part of their shell, and even some bryozoans and some algae require calcium carbonate as well. Now, in order for these organisms to precipitate out calcium carbonate, they need a supply of carbonate ion. Don't worry about the funky letters. Just it's like a thing. Draw a little triangle over it or something. They need free carbonate dissolved in the water and calcium in order to be able to precipitate out the calcium carbonate they need. Now, with the burning of fossil fuel that's been going on for close to 200 years, the atmospheric CO2 has increased. Now, CO2 is very soluble in water and it dissolves readily in the water, but then it reacts with water to form carbonic acid. And as all good acids, this carbonic acid dissociates into a hydrogen ion and bicarbonate ion. Going to take another drink here. Hope I don't remind you of Rubio. Uh, anyway, the measure of acidity of water is based on the concentration of these hydrogen ions. The more hydrogen ions there are, the more acidic the water is. Now, the hydrogen ion reacts with the carbonate in the water and forms bicarbonate ion, which is highly soluble in water. So, with increasing amounts of hydrogen ion in the water, the carbonate ion, the supply of carbonate ions, is being diminished, making it increasingly more difficult for the organisms in the harbor to precipitate out calcium carbonate. So here is that indication of what science tells us is potentially stressful for the organisms in the harbor. And you see that normal is kind of about 8, a pH of 8, and anything below about 7.8 begins to have negative consequences for the organisms in the harbor. And you might be thinking, well, 8 versus 7.8, that doesn't sound terribly different. But recall that pH is a logarithmic scale. So a pH of 7 is 10 times more acidic than a pH of 8. So here's our QA/QC. Whoops. Yeah, QA/QC. Here are the pH data from the Buoy in the 2024 deployment and the 2025 deployment. And here are the data from the NRD's WQAV program. And notice in areas where there is overlap, the WQAV data are elevated relative to the pH data from the Buoy. And for a variety of reasons, again, which we can go into in the Q&A if you like, I am actually thinking that the more reliable data come from the Buoy in this case. So we're going to use pH from the Buoy as a measure of ocean acidification. Now, I downloaded a software tool and it enabled me to input the temperature data from the buoy, the salinity from the buoy, the pH, obviously, from the buoy. It popped out a bunch of other numbers for us. But the main thing is that this software allowed me to calculate what is referred to as the aragonite saturation state. And doesn't that make me seem very fancy? But it's not fancy at all. Aragonite is the form of calcium carbonate that most of the organisms in the harbor rely on and have to precipitate out. And numbers above one mean that the quantity, the concentration of calcium of carbonate, sorry, in the water is sufficient for the organisms to be able to precipitate out. So we have quite high numbers here. So what that means is that in spite of the atmosphere dumping a whole ton of CO2 into our ocean, the aragonite saturation state says that we're still at a level at which the organisms aren't severely impacted by low pH. Um, okay, those are the four problems I want to look at, but remember we were talking about what harbor health means. And what we want to do is link these exposure conditions that the buoy is revealing with how the habitats are behaving. Are the organisms in the harbor increasing? Is the habitat saying stable, sorry. So these data from the buoy and other monitoring programs are only useful in giving us hints about what are the consequences for the organisms in the harbor. And for that, we have to thank the Nantucket Natural Resources Department, who have been conducting eelgrass cover surveys, scallop recruitment surveys for a number of years that will be ongoing, as will the measures from the MMA buoy. So together, we're hoping that all of the sensor activity and all of the ecological data activity can help us have an understanding of what's going on in the harbor. Because keep in mind that the buoy measures chronic as well as accumulating stresses. So the goal is a shared record that links exposure, those variables from our buoys, to outcomes. How are the organisms doing? That is so beautiful. And that is a measure of harbor health. This is extremely important, of course, for Nantucket Harbor. But recall, Nantucket Harbor is a component of the worldwide ocean system. So things that we learn from the harbor can be used to help scientists all over the world think about how we're affecting the ocean. To that end, the Maria Mitchell Association has made contact with and has been invited with the Northeastern Regional Association of Coastal Ocean Observing Systems, which is a clearinghouse for ocean conditions, particularly coastal conditions. And it's a clearinghouse for monitoring programs all up and down the Northeast into Atlantic Canada. And what they have is data from a bunch of different monitoring programs. There's a pull down menu, and you can look at what's going on in Long Island Sound or the Jordan Basin or Massachusetts Bay and so on. And at some point in the near future, I hope that you'll be able to see the MMA buoy data in a pull down menu itself. So I'm really pleased that those of you who came are so engaged in protecting our oceans. I'm grateful that I'm able to work with the MMA buoy and other stakeholders in the area. And let's let's keep it going. Tune in next time, and I'll tell you as our data continue to accumulate what we're learning. Thank you so much.
Thank you, Betsy, for sharing all of your
Yay!
findings and taking
Yay!
questions with us. You did great. You knocked it out the park.
Thank you. Thank you.
Just as a share with our audience this evening, if you have any questions about Dr. Sherman's presentation or even just any general questions related to the Nantucket Harbor or the data that she has discussed this evening, please pop them into the Q&A section, and I'm happy to share and read them out loud for everyone to hear, and we'll get those burning questions answered. But in the meantime, Betsy, I have a couple of questions for you as we wait for people to pop some questions into the chat. So I'll start with high frequency data lets us see patterns that traditional sampling misses. From your perspective, what has been the most consequential invisible event that the buoy has revealed, if anything, about Nantucket Harbor thus far?
Well, the one that is most immediately worrisome is the temperature.
Hmm.
The discrete sampling doesn't catch that,
Hmm.
whereas the buoy has shown that there are continuous episodes of sublethal temperature stress. But I'm confident, given how we're all getting used to the innards of the buoy and how it works, and continuing collaboration with the NRD and other monitoring programs, that as we go forward, we're going to learn lots more and have, I hope, much more predictive power, so-called, what is it, rapid ecological assessment, so that if we see elevated temperatures in the buoy, I might drag a bunch of you out to say, hey, let's go look over here and see what's happening, and look at those chronic stressors over time.
Thank you. All right, we have a couple of questions in the Q&A. I'm going to start with our first guest, Jackie. Shares, I learned today that Nantucket is replacing sand for non-skid conditions with salt. How do you think that will affect buoy data, if at all?
Uh, I don't have sufficient information to answer the question properly. I've not heard of replacing sand with salt.
So this
Uh,
is with regard to, I can give a little bit more context. This is with regard to snow removal and management.
oh, oh, that is a
An
Stowe,
interesting take.
Vermont problem. That's where I live right now,
Mhm.
and I'll tell you, it's terrible for doggy feet. But yeah, anything that goes on the roads will ultimately end up in your water.
Mhm.
And uh, so, to the extent that the town wants to decrease the amount of salt, that is a good thing.
Mhm.
You know, there are two kinds of problems that affect coastal areas, and some are global, systemic problems. The heating of the ocean is happening all over this planet.
Mhm.
uh, the increase in CO2 and the decrease in pH is happening all over this planet. But in addition to these global systemic problems, there are local problems. And for a lot of areas, those local assaults are much more acutely damaging to the ecosystems that surround it. I've done a lot of work in the Cayman Islands looking at their coral reefs, and it is local sewage, runoff, consequences of excessive development that is having the most acute effect on the organisms in the reef. And I would suspect the same thing all over uh, areas of human habitation. near coastal waters.
Thank you, Betsy. I hope that that answers Jackie's question. And just to add, Jackie also said, excellent talk, by the way. Very much
Oh,
enjoyed it.
thank you, Jackie.
Right, moving on to our guest, Peter. Peter says, hi, is the head of the harbor with shallow, less circulating, warmer water has more extreme readings of healthy water systems. How do they compare?
I didn't quite understand the question.
Yep. The head of the harbor with less shallow, with shallow, less circulating, warmer water has more extreme readings of healthy water systems, perhaps. How would you compare that to not the head of the harbor or where the buoy is?
Uh,
I'm ad-libbing. Peter,
okay.
please feel free to add more to your
I
question.
can't hear Peter. Is that correct?
Correct.
Okay. I'll give it a whirl.
Mhm.
Whoop, don't look. That's for another talk. Um,
And he does also add on later, how does the upper harbor compare with Brand Point higher tidal flush?
well, for that, well, here's the solution to Peter's question.
Mhm.
We need these buoys, these moored buoys that are taking constant data, high frequency data, all over the harbor. Um, what we can rely on is looking at the discrete sampling done by the NRD. Um, but it seems to me that because the mouth of the harbor is most affected by tidal movement, it
Mhm.
we might expect conditions in the mouth of the harbor to be less exaggerated than in some of the where that stuff with the don't look, I'm getting to it. I'm getting to it. Doink, doink, doink. Yeah, these guys. So here's where the NRD is taking data. Look, even in these little ponds. So we'll keep using data from the NRD and complementing it with our high frequency data that's here. But it can give us signals for, hey, maybe we should look what else is going on deeper into the harbor.
Mhm. And tell me, and you can elaborate on this perhaps, because the location that we chose for the buoy is it's a very noisy in terms of a data centric focus. It's a very noisy data area. It's a fairly good or could be a fairly good indicator that there are other issues happening in different parts of the harbor, especially if
Absolutely.
there's something concentrated in that in and out flush area. So Peter, if you have more to add to that, please pop it in the Q&A. We'd like to get to address your question a little bit more intimately if we can. Moving on to Amy. You mentioned the MMA buoy being included in the Nirakus, that's what I call them, data
Yep,
in the
yep,
future. Is there
yep.
anything specific rather, that we can do to make this a reality for the 2026 season? Amy asks.
Well, Janelle has already done the heavy lifting in enabling us to link with Nirakus. They need to be able to download our data directly. But if you send me cash directly, I'll see that it goes to the Nirakus project. No, it's on its way. Now the only stopping point is, can I get out of this maybe? So, nope, I don't want to do that. I want to show Nirakus, but what the heck? I'm not going to move slides around. But it's on its way. Janelle pushed the individuals at Nirakus to get us on the list of individual monitoring programs that they are going to link up with. So they have all the information. They said it might be a while because we're kind of in back of the queue, but I'm very excited about that. I would like to mention something, and maybe this is, I don't want to cut anybody off, but one of the things that is true of so many endeavors, but particularly coastal monitoring, it seems that there are lots of programs with slightly different agendas, but with redundant uh, monitoring schemes. And sometimes it feels to me like the left hand doesn't know what the right hand is doing. The USGS has a monitoring program that sometimes I use their data and sometimes I use NOAA data. I wish there were one central uh, clearinghouse for data, but Syracuse is a very important one for those of us in the Northeast.
Also, Amy added that Cash is on the way with a winky smiley face, but she's gonna take care of that for you. We have one more question from an anonymous attendee. What can residents of Nantucket and visitors to the island do to influence any of these measures locally, in your opinion? I know that's a tricky one.
Well, it is, and the first thing I thought of is the ways in which my behavior has a negative impact on uh, climate change.
Mhm,
I like steak. I order from Amazon.
mhm,
Uh, it is the big component here, the big, bad, witchy, awful thing is the burning of fossil fuel. That is the big kahuna.
mhm,
When I was in college, the amount of CO2 in the atmosphere was something like 270 parts per something or other. And I remember, who's that guy from Middlebury? Bill McKibben, who
mhm.
started that 350 organization that we've got to stop the CO2 from increasing beyond 350 parts per whatever. We are now at about 420. We continue to burn fossil fuel, and there has been a push in this country to pull away from uh, fossil fuel burning to other sources of energy. It is being taken up by the rest of the world, but as you all know, there has been a pause on America's leadership in that area. So, in terms of what to do locally, you want to use less fossil fuel by however you do that. I know there's a controversy, I think, abroad about the windmills off the coast. Is that right? And I know that some people are concerned about whales getting banged up. Is that a thing?
You're trending in the right direction, correct?
Okay. Okay. My understanding is that the the problems that whales face as a consequence of these windmills
Mhm,
are small in comparison to what they face because we have big barges moving across our oceans delivering crude oil, and we are making the ocean hotter and more acidic. So right now, I think the data would suggest that you should support the use of these windmills off your coast.
Thank you for sharing. We have two more in the chat. We have Arlene. I'm very much an outsider to this area of research, but if you'll allow this, and yes, we're allowing it, Arlene. Does the buoy measure any toxins that may also be affecting life in the ocean, which is what I always wonder about, Arlene says.
Ah, Arlene is a plant. She is a friend of mine who's got her PhD in biochemistry. Uh, to be alive on this planet now, you've heard about microplastics and PFAS chemicals, those forever chemicals. And the buoy does not have sensors to pick up those toxins. Typically, for those chemical toxins, water samples have to be sent to fancy laboratories to look at those data. I think somebody in the UMass laboratory on Nantucket is looking
Yes.
at microplastics.
Dr. Juanita Urban Rich. I was going to add that, so thank you for
Ah,
highlighting.
ah. So I assume she can look under the microscope, of course, but to
Mhm.
get more quantitative data, I
Mhm.
assume she has to do things chemically in the lab with the microplastics. So you're a biochemist, Darlene. Why don't you develop a sensor that allows us to detect PFAS very quickly?
Arlene, I second that, and I welcome it. Love having another biochemist in the throes, for sure. We have a question from Chelsea. Great, and some commendations as well. Great talk, Dr. Sherman. I was curious if turbidity affects the performance of the sensor.
Oh,
Great
what?
data sets. Go ahead. Or
Yes. Excellent, excellent.
it's a great
Janelle,
question.
we're getting a new sensor. What is it?
It is a turbidity sensor.
Be still my heart. Um, so yes, that's an excellent question. Turbidity affects the organisms directly by, for particularly the photosynthetic organisms, by preventing uh, sunlight from reaching them. It affects so many of the animals by clogging up their gills, not just vertebrates, but invertebrates as well. And it certainly can foul these sensors that are so damn sensitive. So, happily, it seems we are getting a turbidity sensor. Oh, and if you're interested in the data, the NRD on their website, they, I believe, have WQAV data on their website about turbidity, so you can investigate that further. But stay tuned.
Stay tuned, and I can add to that. So, Chelsea, just to elaborate a little bit more, the YSI sensor, the sonde rather, that we have has 5 ports currently on it, and one of the ports is occupied by a central wiper to remediate automatically some of that biofouling for the other 4 sensors. And we determined it early into, our monitoring that turbidity is an essential component that we would like to monitor, but we didn't want to sacrifice or give up one of the other parameters that we've been monitoring. So we were very, very generously funded by the Great Harbor Yacht Club Foundation to expand our Buoy platform from this initial onset of this data monitoring, and we're so grateful for that. And because of them, we've been able to order a larger sonde that has more ports, so 7 ports instead. So we'd be able to monitor 6 different parameters instead. So we're super enthusiastic to get that online for the 2026 season. So as of April 2026, we will be monitoring for turbidity as well and have some baseline data
Great.
for that area. And just circling back to some of the previous questions, in terms of what we can do on the island of Nantucket, I would circle back to Jackie's question and saying perhaps maybe not being more conscientious rather about our output individually and collectively as a community, because everything that we put on the grounds, we put outside, it has an impact on the water system here, whether it's the aquifer or the harbor directly. So just being more conscientious of our, not just our carbon footprint, but just our general overall human footprint is super helpful. And if one person starts, I know in current times it feels really daunting that we are all like, well, what does me turning my AC off for 6 hours, what does that do? And if one person starts, it causes a ripple effect. So it really can make a difference because one person starting influences the immediate family, and then that influences your friend group, and then it becomes a little bit more pervasive in a good way. So I think that that is something that is helpful. And my big thing for 2026 is if we can keep that buoy in the harbor year round, that's a great starting point, but also being able to have at least some triangulated data from 3 different points of the harbor, I think again just builds a more comprehensive idea or illustration of what is actually happening in the harbor from a data perspective. So those are my additions to the questions and to Betsy's responses, if that's welcome.
Absolutely.
Well, given that it is after 8, I had a bunch of other questions, but Betsy, I will always pick your brain and I see you again tomorrow, so I can ask more questions then. But is
Oh,
there anything that we didn't ask you from the Q&A and from the chat
No
rather
one
that we should have
asked,
asked you that you hope that we asked you?
no one asked about my acting nomination, and I'm just stunned. But no, I love the questions, and I hope the next time I talk to y'all, it can be in person so I can see the faces. But I'm very confident that this work is going to go forward. It's robust and in its uh, complementing of other monitoring programs. I'm a tiny bit hopeful.
Yeah. And I would add that it is truly collaborative as well, because we've been able to fold in other organizations and the work that they've already been doing and work that they also hope to be doing, to your point, to complement existing data sets and just build upon it.
Absolutely.
Well, Betsy, I just want to say thank you. And to our audience, I would like to remind us that tonight's conversation reminds us that Nantucket Harbor is not static. It's responsive, sensitive, and constantly communicating. Through the MMA Buoy program, this buoy, this instrument, is helping us to listen a little bit more carefully to see patterns that we can miss with hand sampling or just because human error, and to ask better questions about how we can care for this place that we live in and that we love so much. On behalf of the Maria Mitchell Association, I thank you again, Dr. Sherman, for
Thank
translating
you.
complex data into ecological meaning and for strengthening the bridge between science, stewardship, and community. We're so grateful to have you as our senior marine science advisor and
Thank you so
for
much.
also helping us guide how we understand and protect this harbor that we all love so much, that we're connected to. Thank you.
Thank you.
You're welcome. Thank you.
Night all.
Amy said thank you.
Thank you, Amy. And yep, good.